Jambox is in pairing mode, waiting for device to connect.
Okay, let's get started. So it's a pleasure to have Guy Gaziv with us today. Guy is a postdoctoral fellow in Bar-Ilan's computer vision lab at the Weizmann Institute of Science. This spring, he will be starting his postdoc, I guess his longer postdoc at the Jim DiCarlo lab at MIT. His PhD research focused on the intersection between machine and human vision, and specifically on decoding visual experience from brain activity. This will be the topic of his talk today. Guy earned his BSc, as Bachelor of Science in Electrical and Computer Engineering from the Hebrew University in Jerusalem, and then his master's in physics and PhD in computer science from the Weizmann Institute of Science. We're glad to have you here today with us, Guy, and the floor is yours.
Thank you, Moshe, and thanks everyone for coming to this seminar. Happy to be here. So I'm Guy, a PhD graduate from the Michaeli Group, Department of Computer Science at Weizmann. And I want to tell you today about my recent PhD work. So as Moshe mentioned, my background is actually in computer engineering and physics, and the PhD was in computer science. Let me take you on a bird's eye view of my research. So we wanted to study human vision. And then in the recent years, deep learning has become increasingly popular in studying human vision. And then my research culminated into basically two different branches of research. The primary one is brain decoding, which is the title of today's talk. And this has to do with reconstruction of visual experience from brain activity, where the brain activity is recorded with fMRI. So that's the main branch. And then there is a secondary branch of research that I worked on, which has to do with face representation in the human brain, where we compared representation of faces in human brain, and with that by deep neural nets. And here we considered brain recordings with intracranial EEG. So let's get started with the main branch, and the first branch, which is mind reading, decode images from brain activity. And then the task is as follows. You have human subjects sitting in an fMRI scanner, and they're presented with a bunch of images. And their brain activity is being recorded throughout the session with fMRI. And specifically, we focus on visual cortex fMRI, voxel from area that does visual processing in the brain. And then the question is whether we can reconstruct the image that the subject has seen from those activations, from those voxel activations. So we want to do actually two things. We want to both reconstruct the image and classify it to the semantic category to tell whether it's a goat, a plane, a camel, a fish, and whatnot. And the reason we want to do this is because we believe this is a milestone towards brain-machine interfaces, and one day to develop ability to reconstruct dreams, things that we don't know what the subject has seen actually. And in general, to develop a tool to study consciousness, to tap into information in the brain, be able to retrieve it. And in general, I'm going to talk today about published work currently on bioRxiv and in NeurIPS 2019. So I mentioned that we reconstruct using fMRI brain recording. So let's say a few words about what fMRI is. So here is the brain, and then what fMRI does is it parcels the brain into these small cubes called voxels. And these are, give or take the dimension, given a 3 Tesla fMRI machine. And in each such small cube, you have about 100,000 neurons. And they're firing in response to the visual stimuli presented to the subject. And what you're actually measuring is a proxy for their activity, which is called BOLD, blood oxygenated level dependent signal. This is basically oxygenated blood that is recruited locally as a metabolic process to support these neurons as they're firing. So since you're measuring blood flow, this is a slow process that comes in delay with respect to the electrical activity of the neurons. And in general, you can see that this is poor quality data, low resolution, both in space because you average over multiple neurons in space, and in time because it's a slow process. And also the fMRI machines gives you samples every 2 seconds or so. So this is the data type of data that we're working with to decode and reconstruct the images. So going back to mind reading, we want to cast it as a machine learning problem or a deep learning problem. Namely, we have the fMRI samples and the paired images, the images that are associated with them, and say you compile such a training data. And then you want to build such a model, a deep neural network, that takes you from fMRI, takes them as input and spits out the images on the other side that underlie those fMRIs. And this would be considered as a supervised setting, that you just used a paired training data of fMRI and corresponding images to train such a network. But turns out that this doesn't work well. And why is this the case? Because you have very little data. Okay, you have only about 1,000 pairs in typical fMRI data sets. The subjects cannot spend an eternity in the fMRI. You also have to let them see the image multiple times to have repeats to increase the SNR of your data. And you can't easily combine fMRI samples across multiple subjects because they're different from one brain to another. And as I mentioned, the data to begin with is not very instructive. It's poor quality fMRI data, not single neuron type of recording, which is of very high quality. So you have lack in training data. And in fact, if you are to insist working in the setting of the supervised setting alone, using these 1,000 pairs, these are the types of reconstruction that you'll end up getting. So we see here side by side, the ground truth images that the subjects have seen, and next to it, the supervised only reconstruction, those that you will get if you were to train only in a supervised way. You see that they're quite blurry and don't have much detail to them. And I haven't told you yet how we perform classification to semantic categories, but there's these paired data, the 1,000 pairs of paired data, they cover only about 50 semantic classes in this type of dataset fMRI on ImageNet. And then if you want to perform semantic classification in the style of ImageNet, which is 1,000 ways to fine-grained semantic categories, large-scale semantic categorization, it's very hard to do this when you rely on such limited data. So this doesn't work well as well. So how to cope with the lack in training data? We mentioned the supervised training approach, and here is what we propose. So we propose to also consider the inverse direction of the mapping, namely decoding is fMRI to images. Now we consider also encoding, which takes us from images to fMRIs. And why do we do this? Because using the other direction, we can add self-supervision in the following way. We take the encoder and the decoder, we put them back to back. Now the input of the encoder is an image, and the output of the decoder is also an image. And if the encoder and decoder are trained well, this should be the same image. It's just cycle consistency. And there in the middle, between the encoder and the decoder, there is pseudo fMRI, but you don't need to assume knowing anything about it. You can just train here on images that you don't assume knowing anything about their fMRI recording. These are just natural images from ImageNet, say. And this is what we did. We took additional 50,000 natural images from ImageNet without fMRI recording whatsoever. And we trained it in the following configuration. And what you virtually get is adaptation to statistics of natural images. Now, even more than this, you can also cascade it the other way around, the encoder and the decoder. Now the input of the decoder is an fMRI. And the output of the encoder should be the same fMRI. Again, cycle consistency only in the other way. And now you can train on more fMRI data than what you originally had in the paired training data. And this can be fMRI that you assume nothing about the image that is paired with it. So for example, you can take here test fMRI. And namely, these are the test fMRI that you in the end want to reconstruct images from, but you don't assume knowing anything about the image. So you don't do anything bad with the task that you're trying to perform. You just trained on the input test data. This is also called in machine learning transductive learning, where you train on the input test data. You basically adapt the decoder to the statistics of your specific test data, other than trying to build a decoder that generalizes very globally to any type of fMRI data. And it turns out that actually the first criterion of the two, the encoder-decoder, is extremely powerful, especially when trained with a specific type of loss between the input and the output images. I'll show you what happens when we train with perceptual similarity loss. Then it becomes so powerful that it even marginalizes the effect of the second criterion of training on the test fMRI. But altogether, this addition, yeah?
I might be missing something. If you only have the self-supervision, then any encoder, which is just the inverse of the decoder, that would work fine, right? It could be the identity. So you do need like a large weight to the supervised training part.
Correct. So definitely, I'm not talking here about the method in detail. This is just conceptualization. I will get into the details of the method, and then you will see how this doesn't collapse to identity. So we don't train it as a just autoencoder. We train it in phases, where we first train the encoder, and only then we train the decoder, such that the phenomenon that you described doesn't happen. I'll get into the details of that. So this is just conceptualizing how we add self-supervision as a way to enrich the training data. And let's see the effect of this addition. So here are the supervised only reconstructions that we have seen before. And now next to it, we see what happens when we add self-supervision. We see that we gain a lot of details in the reconstruction. They look now much better. Some colors look better, and also texture and details of the original objects. And this is one data set called fMRI on ImageNet. And here is another data set. So the fMRI on ImageNet is by the Kamitani group from Kyoto, and VIM1 is a popular data set by the Gallant group from Berkeley. And we see that in both cases, we add detail to the reconstruction beyond what we had before, and we're gonna survey more reconstructions later on. But that's the addition of adding self-supervision, the added value of it. And now back to the method, Gal. So we train in two phases, okay? So we start by training the encoder in a supervised way. Namely, we take the paired training data of images and fMRIs, these 1,000 pairs, and we train the encoder only. Then we fix the weights of the encoder, and we use it to train the decoder in a second phase. And in the second phase, we train on 3 objectives simultaneously. We have the supervised training, where we have samples of fMRI and images, and we train on the objectives of training on unpaired natural images and training on unpaired test fMRI data. So in each batch, we have composition, it is composed of all of them. So we have supervised examples, unsupervised examples of images, and unsupervised examples of test fMRI. And then the loss ends up being a sum of the 3 components of the loss. Each of them is normalized separately to verify that none of them is being too over-dominating the others. Yeah, and then I mentioned that this specific criteria, yeah?
So allow me, for the discussion. So the surprising thing here is that the encoder is trained only using the very limited supervised data you have. So in a way, you're saying that representation is flexible enough so the decoder can do enough, can correct all the damage that the first phase does?
Right, so that's an interesting point here, that there is basically, it's not very symmetric, right? So we say that the decoder has to benefit from unsupervised objectives too, but the encoder trains only in a supervised way. We also looked into what happens if we do in iterations, where after training the decoder, we then train the encoder also in a supervised way. Turns out that it saturates very quickly, and there is not much gain to it beyond just having it like that. And another argument for the lack of symmetry, which actually drove us to originally develop this method, that we realized that there is something easier in doing encoding, doing good encoding than doing good decoding. So decoding is tough. The fMRI is about 5,000 numbers, 5,000 voxels. And then the image is about, if you think about it, 40,000 numbers. So you're trying to arrive from very little data to a lot of data, from 5,000 numbers to 40,000 numbers. Turns out that this is quite hard. Whereas in the other direction, going from images to fMRI and doing it in a good way is slightly easier in our experiments. And then this is the idea that you actually focus on what's easier for you first. You train an encoder, and then you use it as an auxiliary model to train the other model, which is harder to train. Was that answering your question?
Yep.
Yeah, cool. Yeah, so I mentioned that this criterion is particularly powerful, training on additional unpaired images. And here is why. We train it in using perceptual similarity loss. This was presented in CVPR 2018. And this loss basically takes the input and the output images, and it constrains them to be similar in multiple levels of a pre-trained classification network. So they should be the same both in low-level features as well as in high-level semantic features using a cosine similarity. And this should basically align more closely to how humans perceive a similarity of images. And what's nice about it is that once you train on 50,000 additional natural images from 1,000 rich ImageNet classes, you basically gain here adaptation to novel semantic classes beyond what you had in the paired training data. So you never assume knowing anything about the semantic classes of images. You don't use the labels. But then once you have an image and an output image and you constrain them to be similar semantically, you gain some knowledge in your decoder about semantics beyond the 50 categories that you had in the paired data. So you adapt to new semantic categories. And this is, so the perceptual similarity loss is useful both for the better reconstructions and to increase the performance of semantic classification, as I'm going to show. So now let's delve into the reconstruction themselves. Let's look at the entire test data. So this is fMRI on ImageNet, image shown to the subject. And now we see the reconstructed image from test fMRI data. Never assume knowing anything about this image. And here is another example of this whale. And this is reconstruction from test fMRI. And here is the plane. And we see that in the snowmobile, we see that in many of these images, we have very accurate details of the original object. Sometimes it's more identifiable, sometimes less. Sometimes the colors match better the ground truth, sometimes less. And actually there is lots to study about this beyond what we did to realize when do certain stuff being reconstructed more easily and when less. It also depends on the specific subject. Here we see it from a single subject. So for other subjects, the better reconstructions might be others. So there's lots to study in this, but these are state-of-the-art reconstructions actually for this data set. And here is comparison with other state-of-the-art methods. So we see here method by Shen et al. So this is from the Kamitani group that created this data set. And they incorporated natural image priors. So their method for handling the lack in training data is to constrain the reconstructions with natural image priors, with GANs. And we see that their images do have a natural image appeal, but they're not as faithful as ours with respect to the ground truth image. And when we evaluate ourselves against them, we outperform them. And here's another method by Sentivest from the Nacellaris group for the other data set, which is also, by the way, tailored. Each of these methods was tailored for a specific data set, either fMRI on ImageNet or the VIM1 data set. And we were the first to actually propose a method that works well on two data sets at least. So we see also visually here that our reconstruction for the VIM1 data set, the grayscale one, outperformed the previous state of the art there. And then how do we evaluate this, right? Because I'm just hand waving here, showing you that this looks better. But then a way to evaluate this is by n-way identification. And this is how it works. You take a reconstructed image on the top here, the goat, and then you take candidate ground truth images for it. One of them is being the true ground truth image, here this goat, and the rest are randomly selected images. And then this is a two-way identification setting. And now you take a metric, a distance metric, say the perceptual similarity or a human annotator, and you ask it to say which of the candidate ground truth images is the closest to the reconstruction. And then if you get that this is the closest, you say correct classification. And then in this example, if it says that this is the nearest neighbor, you say incorrect classification. And then you accumulate such statistics. And this is what it looks like. So on the top, we see results, comparison of fMRI on image at our method with respect to two variants of the Shen et al. Method with a prior and without a prior. And we see that we outperform it. This is two-way identification. And also for the other data set, the VIM1 data set, these are these reconstruction compared with these, and we see that we outperform them. Dashed line corresponds to chance level, right? 50%. But then also as you scale to 5-way, 10-way, 50-way. Now 50-way means that you have 49 distractors, one being the true ground truth. So we managed to do it quite well with our method, and the chance level is 1/50. So it shows the same picture that over n-way identification and across the two data sets, we outperform state-of-the-art methods. And this was based on perceptual similarity metrics. So the way that we compare the reconstructed image with each of the candidates as by perceptual similarity. But then, as I mentioned, you can also let a human annotator do this for you, right, in a behavioral survey. So this is what we see here. So we outsource it to Mechanical Turk, and human annotators annotated it for us. Each time they said which one is the closest, and we see again the same picture that we outperform the other methods, which makes us the current state of the art in image reconstruction from fMRI. But then I mentioned that we also do classification, right? This was all reconstructions. How do we perform semantic classification? So our method is as follows. We take an fMRI and we feed it forward through a decoder and reconstruct the test image. And then from this test image, we extract deep features using a pre-trained classification network. And then we contrast this signature against a gallery of class representatives. What are these class representatives? So you take 100 random images of, say, tiger shark class. Each of them you feed forward through a pre-trained classification network, you extract the features, and then you take the average feature, you call it the class representative of tiger shark class. And you do this thing for 1,000 classes, ILS VRC channel challenge. And then once you have these extracted features of the reconstructed image, you can then rank or sort the gallery from most similar to least similar and say which is the correct class label for this image. And these are the results. And mind that the images here don't matter, they're just for visualization. We actually show here the images that are the nearest neighbor within the class representative, but what truly matters are the names of the classes on the top. So we see here ground truth image, and then reconstructed from fMRI. And then these are the top 5 predicted class labels out of more than 1,000 classes. And we see that the first is indeed the housefly, as the ground truth is, which is very nice. But even nicer than this, we see that the others are also semantically very reasonable and relevant. We see here insect, cicada, fly. So that's very nice. And here are some more examples. Each of them shows the top 5 predicted class label out of more than 1,000. So airliner, indeed, the first is airliner, but then the second is a warplane, which is a distinct class in ImageNet. Yeah, and the others are types of vessels, which are also semantically not very far off an airliner, I would say. Stained glass window, the first is stained glass window, and then vending machine, slot, altar, which has some shared semantics, I suppose, with stained glass window, beer mug, with all these mugs and coffee pots, and kitchen-related stuff. And here with the swan, the first one is not the swan, it's a duck, which is, again, semantically close to swan, but it's a different class. The fourth is the correct swan. So we see in all of these correct classification cases, namely that we have the ground truth class among the top 5 predicted class labels. In all of them, we see nice semantics even in the non-ground truth labels assigned. And then how do we evaluate this? So we follow the same logic of the 50-way identification, this time for classification. So you have the class represented, the deep features of the reconstructed image, and then you contrast it with the ground truth class representatives and randomly selected class representatives. And so here 50-way, so chance level is 1/50, and we see what happens when we take the self-supervised reconstructions and we do it, contrasted with what happens if these reconstructions were the supervised only reconstruction. So adding self-supervision clearly helps us do this semantic classification. And it works even when we scale up to 1,000-way. Now, 1,000-way means that the gallery is of 1,000 examples, and you do really a 1,000-way classification. So the chance level of getting the right class is 1/1,000. And we do 100 times better than this, so 10%, which we were very proud of this, because if you were only to use the paired data, it covers so little classes, so it's extremely challenging thing to do. Yeah? Question? Yeah, so we were very happy about this, but then we asked, like, what happens with all the other cases where you don't manage to get the correct class label? And these are incorrect classification cases. So we see here the ground truth domestic llama, and here the top 5 predicted class labels are all these white fairy dogs. And here with the leopard, you got a lion and other fairy dogs with similar type of fur. And here with the camel, you got Arabian camel, which is not a camel according to the categories of ImageNet. It's a different category, but clearly there is very nice semantics being extracted here, the top 5 predicted class labels. So that is to say that our measure here is very conservative. Even if in incorrect classification cases, you still get very nice semantic classification. So that concludes the reconstruction and classification. And then we asked things more related to the brain side of things. For example, the reconstructions consider the entire visual cortex, but where is it coming from? Is there a specific region that is should be getting most of the credit for the quality of the reconstruction or not? And I'm just going to go very briefly on this. So these are the full visual cortex reconstructions that we've seen before, and these we accumulate over the area. So this is V1 only reconstructions. We see that they're already getting a lot of the full visual cortex reconstruction details. And this is what happens when you take V1 to V3. Already almost all the details are there, even though it's about half the number of voxels as in the full visual cortex. And here is what happens when you consider only the higher visual cortex, lateral occipital cortex, fusiform face area, parahippocampal place area. So this is about the same number of voxels as in the LVC, but the reconstructions are not that good. So they don't add a lot actually to our reconstructions. So the quality is dominated by early visual cortex. Another thing that we wanted to ask is whether the models capture any properties of the brain. How do they do this magic of the reconstruction? And then for this, we looked at the encoder and we looked at the modeled voxels at the output here. We wanted to ask what is their preferred stimulus. So for this, we perform activation maximization over the input image to drive to maximum each specific unit here. And then we get these receptive fields of the model voxels. We already see that they're localized in space. They're not all over, which is nice. This is what you would expect from a receptive field of a voxel. And then the next thing we did is to measure where is it in space and the radius. And then for each voxel, we can plot it on the cortical sheet, right? And this might strike some of you as very familiar because this is a known retinotopic mapping in neuroscience. What we see here in the polar angle plot is horizontal to vertical meridians transition. Namely, we have here blue, which is the horizontal meridian, being translated to green, which is the vertical meridian, back to blue, back to green. And also here in the bottom, we have blue to red to blue to red. And also that you have the inversion, what's been in the lower part of the visual field is being translated to the top part in the brain and vice versa. And also here in the eccentricity plot, you see that what has been in the fovea area of the visual field is being translated to foveal areas in the brain. What has been in the periphery is being translated to the periphery here. And to put it in the context of a neuroscience paper or reference, so here is retinotopic mapping by this reference, and we see how similar this image looks, these transitions from red to green, from blue to green to blue to green. And this totally spontaneously emerged from training on data. We never enforced anything about the receptive field of any of these voxels. So actually they capture something that has to do with what you would expect from biology. And before concluding the reconstruction part, I just want to mention our very recent work where we actually extended reconstruction to also reconstruct depth maps. So before we reconstructed from fMRI only the RGB images. Now we were able to also reconstruct depth. We call it more than meets the eye, because now we reconstruct things that the subject never met the eye of the subject. Subject has seen only the images, but now we claim that we can decode additional layers of information that are encoded in his brain that are interpreted from these images, and we can also decode them. And this is, by the way, due to the self-supervised approach. This enables us to do this. I'm not gonna detail on the method, but just to give the flavor of things. So these are the reconstructions of RGB, similar to what we've seen before, ground truth images side by side with reconstructed from fMRI. But now we also have reconstruction of the depth maps. So we reconstruct RGBD. And we see the nice depth maps side by side, ground truth depth maps that are estimated using a depth estimator network. So we hope that with this, we can possibly expand in general the decoding of information to new types of information beyond the pixels of the image, say bounding boxes, semantic segmentation, and other stuff. So this was the brain decoding works. And now let me switch gears and tell you just very briefly about face representation in the human brain, our work on comparing neural nets and representation in the brain with electrodes. This work was published in Nature Communication 2019. And what happens in this task is that there are clinical patients with electrodes implanted in their brain for other purposes, for clinical interventions. And then these subjects were presented with face images from 3 sets. And they were tasked to do a one-back visual task to say whether they see the same image twice in a row. And then you have these representations with electrodes, a few dozens of electrodes in their brain. You have representation of those images. And then the question is, say you take, and these electrodes are from face-selective areas from FFA. So they respond specifically to faces. So these are areas dedicated to face recognition. And then say you take another artificial counterpart, a pre-trained face classification network. And then you want to ask whether they have something shared in how they represent faces. And specifically, there has been a hypothesis about face space geometry, namely how pairs of faces are represented in the brain and the same pairs are represented in the network, whether they have the same face space geometry. I'm going to explain in more detail what do I mean by the pairwise similarity structure. But we were looking for similar representation in both the brain and the network. And this is what I mean by pairwise similarity or face space geometry. So subjects see the images and then they have a brain response. And you take the same image, you feed it forward through a pre-trained classification network, you get the response of the network. And then you take another image, you let the subject see, you have response, and you have a response in the network. And now what you do, you compute the delta, some distance, Euclidean distance between the response of the first image and the second in the network and in the brain. And you can create such many deltas. So each pairs of images create you new delta, both in the CNN and in the brain. And then when you have all these deltas, you can ask whether it's the same delta, whether they correlate strongly. This would mean that the brain and the network have the same face space geometry. So here is another view of this. So here are all the possible deltas in the CNN for all these images and in the brain, and then you just straightforward perform the correlation. And what's nice about this work is that we found very, very strong correlation that is basically unusual in neuroscience and in specific, with specific layers in the network. So we see here very strong correlation with layers that are intermediate in the network. They're not the top layers that are known to be more agnostic to low-level details and are more tuned to the identity of subjects. And these are not also the layers that are very tuned to low-level visual details. They're somewhere in the middle. Namely, they have some sensitivity to pictorial aspects of the image, but also some invariance to it. So, and this was very striking. We found it for 3 different data sets, pretty much the same picture that you have strong correlation in the middle of the network in these layers. And this is for aggregated across the 3 subjects. So that's the picture. And you also see contrasted here with what happens when you don't train the network at all. So it's important that the network is pre-trained. Yeah, so once we establish this connection that there is strong correlation with these layers in the brain, we wanted to ask what happens if you, what do you need to do to abolish this correlation? So now from the subject, it's already set. Now what happens when you do manipulation to the images and present it now to the network? So say you take a profile of the original images, a half profile or a full profile, well, this abolishes the correlation. But if you only remove background or change some luminance level or move it to grayscale, then it doesn't abolish the correlation. And another thing we did was to take specific artificial units that are very correlated with electrodes in the brain, and then to ask what is their receptive field, and to imply from this on the receptive field of possible for the electrode, for the biological electrode or area. And we found that the receptive field corresponded to high-level features that you would assume are relevant for identity recognition, for face recognition. So things like an ear or an eyebrow, but not things that capture the entire image or the entire identity. And also notice that these things, these receptive fields are sensitive to view. So if you were to change the view or pose of the face, these things would change too. So altogether, our findings support a pictorial function of face-selective regions of the visual cortex. So unlike a hypothesis of a grandma cell that only responds to the identity no matter what, no, it is also tuned to pictorial aspects, and it's somewhere in the middle between low level and very high level. So these are the 2 branches of my research. I talked about the primary brain decoding and the face representation. And if I conclude the take-home messages, so I've shown self-supervised approach for image reconstruction and semantic classification, state-of-the-art reconstruction and unprecedented large-scale semantic classification, predominance of early visual areas in reconstructions and biologically plausible models, also the RGBD depth reconstruction. And in the face representation, I've shown you convergent evolution of face-space geometry across humans and neural networks, and the pictorial role of high-order face-selective regions in the visual cortex. And these are all my collaborators, most of them. At the top, we see the first authors on the papers, Roman Bailey, Niv Granot, Shani Grossman, and more co-authors, Asaf Hugi, Francesca Strappini, Tal Golan, and of course, my dear supervisor, Michal Irani, and Rafi Malach with the face representation work. And I just want to spend a minute to tell you about my future path and hopes. So this was my path until now during my PhD. And now for postdoc, I'm looking forward to join the DiCarlo lab at MIT. And the bread and butter of that lab is biological models of primate vision. So if many of you have heard of brain score, where we score models according to how well they predict brain activity. And then one important question that comes to mind very often is, why do we need very accurate models of biological vision? What can we gain from this? And I think one answer to this is neural control. Namely, if this was brain decoding with neural control, you can actually cast desired brain states on a brain in a non-invasive way. You can just prompt your model, probe it, and ask it, what do I need to do to a visual stimulus? Just how do I need to synthesize it in order to induce a desired brain activity? And they already had a work along these lines, neural population control via deep image synthesis, work and published in Science, where they synthesize these stimuli and they managed to drive V4 neurons to desired brain activity, even outside the natural range of activity for these neurons. And I think it holds great promise for all kinds of clinical applications. Many of us spend a lot of time on the screens today, and imagine that you could induce slight modulation onto existing stimuli and say drive your amygdala higher or lower and gain some positive clinical effect from this. So I'm looking forward to do these things. And with this, I want to conclude, and thank you very much for your attention.
Thank you very much, Guy. Before others will unmute themselves to ask questions, let me just ask you a technical question. Can you tell us more about how you select the specific voxels from which you encode and decode?
Yeah, so basically these are visual cortex voxels, and your way of doing this to decide what's a good visual voxel, basically retinotopy you have, right? The retinotopy experiment. And then you also do, you have multiple repeats of the same image, and you say a visual voxel is such that responds the same to different repeats of the same image, and is not the same for different images. So this is basically your selection criterion. You want it to be the same across same presentations of the same stimulus.
Okay, if there are any questions, you guys can unmute yourself and just go ahead.
Can I ask you something?
Sure.
So so there is a time delay between the time that the picture is presented and the time that you have the response in the fMRI. Do you adjust for this difference?
Right, of course. So this is a block design experiment. You basically perform common pre-processing methods such as GLM to arrive from the continuous time course of the fMRI to have beta coefficients that are basically the per discrete stimulus activations in the brain. In video, actually, what you mentioned is much more critical. You have to be very tuned to synchrony aspect. It's much harder to do. We also do video reconstruction. There, it's much messier. Also, the stimulus changes rapidly. You have a high frame rate. Here, the stimuli that you've seen are for the main data set, fMRI on ImageNet, they're presented for 18 seconds. Sorry, they're presented for 9 seconds and they flash at 0.5 hertz. Namely, there are 18 flashes of each such image. And then you have about 1 second delay between this image and the next image block. So this is what the experiment looked like and how you derive the image-specific responses, the beta values.
I have a question.
Yeah, sure.
Hey, Guy. Thank you for this very interesting talk. And I'm very impressed by the decoding you showed. But I'm going to ask you something which I've also asked Shani and Rafi. Are we really learning something about how the visual system works here by having these parallels with how the DNNs and RNNs work? I mean, are we really learning something about how the visual system in humans or in primates really works?
Yeah. So I think from what we presented, not directly, it's more of a milestone towards it. It introduces a new tool to the community that with this, you can ask new questions. And I think for this, it's important, as I mentioned, especially to study consciousness. So for this, you need to be able to tap into information in the brain. And on the other hand, it can be potential basis for prosthesis. And brain-machine interfaces. So I think it mainly aims to these aspects. And currently, we don't directly have any scientific gain on how visual processing works.
Okay, thanks.
Okay. Well, thanks again, Guy. It was indeed very impressive in scope and level of performance. Good luck to you also at MIT.
Thank you very much. Thank you very much. Pleasure.
Great.
Thank you, guys. Very nice.
Thank you.